What's up, y'all? Welcome to another conversation in Purple Bear Biology. In this episode, we are going to discuss how our bodies maintain homeostasis and how chemicals in our body interact. So let's jump right into some slides and talk about the chemistry of anatomy. Homeostasis is the stable state that our bodies must maintain to function correctly. There are numerous variables that our body must control in order for cells to survive and thrive. You already know many of them that are listed here. These variables have values that are ideal for cellular function called set points, but often have ranges that the variables can fluctuate within and our bodies can still function. The overall range of fluctuation that is acceptable depends on the variable that is changing. Some variables like temperature can change a few degrees Fahrenheit and the cell can still function correctly. But other variables like blood pH can only fluctuate minute amounts before causing systemic problems. So how does our body return to homeostasis when our external environments change our internal ones? Our bodies use something called feedback loops. There are two primary types of feedback loops, positive and negative. While negative feedback loops are the primary mechanism for returning our body to homeostasis, positive feedback loops occur during unique scenarios that do not return our body to homeostasis. Both feedback loops start with a stimulus or a trigger that initiates the loop and gets a response by our bodies. In negative feedback loops, our body's response to the stimulus reduces the environmental stimulus that is driving our bodies out of homeostasis in an effort to return to our set point. Let's use an example to clarify this concept. Let's use a room with temperature control as our first example. Imagine you set a thermostat in your living room to 20 degrees Celsius, about 68 degrees Fahrenheit. As you hang out in the room and use electronics, the temperature of the room begins to rise. The thermostat recognizes the change in the temperature and turns on the air conditioner to return the room to the set temperature. This is a negative feedback loop. The stimulus is the rising temperature. The response is the AC turning on. So what happens when the room reaches the set temperature? The AC turns off. The same thing happens in our bodies with negative feedback loops. Once homeostasis is restored, there is no longer a stimulus to initiate the response, and the negative feedback loop shuts off until the measured values increase or decrease again. Does the feedback loop work the same if the temperature of the room got colder instead of hotter? Sure. The loop still operates the same, but the response changes because the stimulus is different. So how does this work in your bodies? Well, it's actually pretty awesome. A region of your brain called the hypothalamus monitors your body's temperature. If your body gets too cold, the hypothalamus triggers shivering to generate heat and constricts your blood vessels in your arms and legs to conserve heat near your vital organs. However, if your body temperature begins to rise, then your blood vessels dilate and you begin to sweat. And this is just one example of numerous negative feedback loops that your body coordinates continuously to maintain homeostasis. Okay, so if that's how negative feedback loops work, then how does a positive feedback loop operate? Positive feedback loops are less utilized and often activated during unique scenarios that the body may find itself in. In these types of loops, the response to a stimulus actually takes the body further out of homeostasis. A good example of this is blood clotting. Let's imagine that you cut your finger with a knife while cutting carrots for dinner. Losing blood can be disastrous by changing your blood volume and blood pressure, so our body will quickly try to clot the site of damage. The way that this works is that cells in your blood called platelets stick to the site of damage and then release chemicals to recruit more platelets to the area. What happens when new platelets arrive at the site of damage? Well, they also stick to the site of damage and the other platelets that are already there. Then they release chemicals to recruit more platelets. The process continues until the cut is filled in and a plug is formed. When no more damaged tissue is exposed, platelet recruitment is terminated. Now, two important things to note about this type of positive response. First, the response to the stimulus increased the stimulus itself. Damaged tissues recruited platelets, and those platelets recruited more cells to the site. This is the exact opposite of a negative feedback loop where the response reduces or eliminates the stimulus that activated the loop in the first place. Second, because positive feedback loops respond to stimuli and increase the body's reaction, these types of loops must be self-limiting or self-terminating. What does that mean? Well, imagine that the platelets never stopped recruiting more platelets. Eventually, they would block the entire blood vessel and not just the cut. That would be really bad for blood flow. So positive feedback loops, like blood clotting, must have the ability to shut themselves down when the response is no longer needed. Alright y'all, so now that we have some understanding of what homeostasis is and how our bodies maintain that stable state, let's shift our conversation to some chemistry behind homeostasis. Our body is a composition of different chemicals constantly fluctuating. While this chemical composition can vary from individual to individual, the major chemical profiles are pretty similar between all of us. Our body is mainly composed of 11 different types of elements depicted here. While the relative percentages are listed, do not make the mistake of thinking that those that occur in low percentages are less important. Many of these elements that occur in relatively low concentrations have a profound impact on our body's ability to function correctly. All of these elements interact to form compounds throughout our body. The interaction between atoms of each element is referred to as a bond. You can think of this a lot like Legos, where each atom is a Lego and they stick together to form larger structures. What causes the atoms to interact? Electrons. Atoms are composed of subatomic particles called electrons, protons, and neutrons. While protons and neutrons are found at the center of the atom, the electrons orbit around them. Aside from the first orbital layer that houses 2 electrons, atoms want to have 8 electrons in their outer layer around them. If we look at this carbon atom, it has 4 electrons in its outer orbital. So the carbon atom would like to have 4 more electrons. Where can it get those electrons? Well, it can share those with other atoms. When atoms share electrons, they form what is called a covalent bond. This allows all of the atoms to obtain a full electron orbital. I should mention that not all atoms learn to share fairly in kindergarten. Some of them like to hold on to electrons that they are sharing, a lot like a ball hog on a basketball court. When atoms like oxygen do not share the electrons evenly, they create partial electrical charges at their poles. So we call these types of covalent bonds polar covalent bonds. These partial charges on oxygen and hydrogen in water molecules create electrical attractions called hydrogen bonds that are attributed to why water cohesively sticks to itself. This hydrogen bonding accounts for numerous other properties that influence the physiology of water as well. We'll get the opportunity to discuss all of those later in another video. There is one other type of bond I want to discuss: ionic bonds. Sometimes atoms only have 1 or 2 electrons in their outer orbital, but have a full set of 8 in the next orbital down. The chances that they will be able to make 6 or 7 additional covalent bonds are pretty slim. So the atoms often give those electrons to other atoms. But electrons are negatively charged, so if an atom gives up some of this negativity, then it becomes positively charged. Additionally, the atom that receives the electron becomes negatively charged. These newly charged atoms attract to one another and form a charged bond called an ionic bond. While these chemical properties may make you want to run and hide, they are vitally important to understand how and why chemical compounds in your body combine or break. They determine how other molecules interact with our cells, as well as how molecules get transported throughout the body. They also determine how energy gets converted and transported from one form to another. Okay, so let's summarize what we learned in this discussion. Our bodies must maintain a stable state called homeostasis to survive and thrive. Negative feedback loops allow us to respond to stimuli and return to homeostasis. However, positive feedback loops allow our bodies to respond to stimuli, but take us out of homeostasis and must be self-limiting to avoid disaster. To maintain homeostasis, our bodies must balance the different chemicals throughout us. Chemical compounds are built from atoms that interact based on electrons. Covalent, hydrogen, and ionic bonds all describe how molecules interact based on electrons and charges. Whew! That is all pretty cool and is going to serve as the foundation for our next couple of conversations about chemistry of anatomy. If you found the video helpful, hit the like button and subscribe. Drop a comment in the comment section below if you have any questions or want me to discuss more about any of these topics. Thank y'all for stopping by, and see you next time on another edition of Purple Bear Biology.